today to introduce to you all Adam Martini. He's a professor at University of California, Irvine. He got his master's and doctoral degrees from the Technical University of Denmark, after which he went to MIT and did a postdoc studying marine microbiology. And he's been at UC Irvine since 2006, where he's ascended through the ranks from assistant to associate to full professor as of 2017. Adam's research has been really remarkable in my view. And I can say this because really, when I want to follow the field of biological oceanography, at least from my perspective of it, and the role of microbes in the functioning of the world's oceans, I find myself over and over again turning to his papers and using them in my classes. The aims of his research is really to understand how microbes respond and adapt, often genetically, to different gradients in the world's oceans. And also based on that adaptation, how different microbial communities organize themselves and how the biodiversity of microbes in the ocean is a result of their ability to adapt or not adapt. And of course, these sorts of understandings play a critical role in understanding the role of microbes in global elemental cycles, and also in predicting how our oceans are going to change in the face of climate change. And his papers are, you'll find them across all of the top journals: Nature, Science, all of the Nature in betweens. I think close to a dozen PNAS papers. And and I saw his talk at ASLO this summer, and I said to myself, it'd be great to get him to speak to us here at the School of Marine Atmospheric Sciences, because just when I thought he had his greatest breakthrough in a science paper published in the spring of 2021, then he presented something entirely different at the ASLO meeting, which I thought was equally or even more fascinating. So I'm excited to see this talk, and I think it's going to be very useful for our students. So without further ado, I bring to you Dr. Adam Martini.
Thank you, Chris, for that very kind introduction. And I'm very excited to present my work to you, and hopefully have some good conversations at the end and afterwards. So today I'm going to talk about some new things that we're working on, as Chris alluded to, and it's really about how we can use microbes to inform us about how the ocean functions, and also start to detect maybe perhaps subtle changes in ocean processes from in ways that might have been challenging to do just with sort of chemical or physical measurements alone. In other words, this idea that we can use microbes as sort of a living biosensor for ocean changes. As Chris said, I work at University of California, Irvine, and most of the work, here we go, are done by members of my lab or close collaborators that sort of sits down the hallway. In particular, I would like to highlight my postdoc, Elise Larkin, and one of my grad students, Lucas Eustick, who's done a lot of this work. But really, what I'm going to present today is sort of the culmination of a last decade worth of going to sea, collecting a lot of samples, and then sort of a convergence between some of the molecular biology work we've done in the lab and our understanding of changes in the field and ocean biogeochemistry. Lastly, I'd like to acknowledge NSF, NASA, and NOAA who are funding this work. So what limits ocean productivity? This is sort of a centennial-long question that has been the topic of much debate. And I will say that there are many different opinions to the answer of this question. The classic view on what limits ocean productivity is that surface phytoplankton are generally limited by nutrients. not in all places, but in many places. And the nutrients that are most commonly biolimiting are nitrogen, phosphorus, and iron. And so the classical view on whether it's nitrogen, phosphorus, or iron considered to be represented by this leaky barrel. I assume that some of you have seen this idea before, but in general, it is that the resource that is in the lowest supply relatively to the demand of the community is the one that's going to be limiting. So in this leaky barrel, the piece of wood that is the lowest is where the water is going to flow out. And so in this graphic, nitrogen would be the limiting resource. This is also called the Lubix Law of the minimum. And the idea is that there's sort of one primary limiting nutrient, that there has to be one element that is sort of in less supply than the rest. From sort of a variety of different perspectives, N is thought to be the primary limiting resource in most of the ocean. And that's probably because there's also denitrification occurring in the ocean. So eventually the nitrogen will be somewhat depleted relative to something like phosphorus. One of the primary tools that people have used to understand nutrient limitation in aquatic ecosystems, whether they are marine or freshwater, are these nutrient addition experiments. And this is also a popular tool in oceanography. So as you can see on the map, people have gone to sort of a variety of ocean environments, although as you can also see, it's a very non-random distribution. And they scoop up some seawater, add various combinations of nutrients, and then look for which combination stimulates growth. And the colors represent the inner circle is the primary limiting nutrient, and the outer circle is the secondary limiting nutrient. And so there's a couple of take-home messages from this analysis that was done a few years back. First of all, you can see that from sort of 40 north to 40 south, most of the dots are green in the middle, meaning that there's mostly nitrogen limitation. If you go to sort of higher latitudes or in the eastern equatorial Pacific, these are places where there's residual nitrate in the surface water. And there you tend to get iron limitation represented by the red dots. Then also that there's some P co-limitation, maybe in the North Atlantic Ocean, represented with the black outline. And then also that there are many places where there's some co-limitation, meaning that the colors of the inner and outer are different. But in response to this paper and another paper by Jim Elser that also came to this conclusion that the co-limitation was widespread, Eric Davidson wrote a response to that saying that, well, maybe there's a lot of co-limitation, but an alternative hypothesis is that in many aquatic ecosystems, nutrients are present in very low concentrations. So when you add elements, you quickly run out of even non-limiting elements in the sample. So for example, in the oligotrophic gyres of the ocean, you have maybe a couple of nanomolar of phosphate and a couple of nanomolar of nitrate. And so no matter what you add, if you add nitrate, you're going to run out of phosphate. And if you add phosphate, you're going to run out of nitrate. And so only when you add nitrate and phosphate do you stimulate growth. And so this idea of co-limitation could also just be the quick expulsion of non-limiting nutrients. And so there's a lot of other reasons for why there are issues with these bioassays for detecting nutrient limitation. And that is because nutrient stress is a complex thing. Because when a resource becomes limiting, communities will respond at a variety of levels of biological organization. And so, for example, you might see some evolution whereby organisms adapt to a low resource supply by improve their nutrient uptake affinity, for example, overexpression of transporters. They might access alternative forms, like organically bound nutrients using an enzyme like alkaline phosphatase. Or they might be frugal in their use to lower their demand, for example, through a loss of function that requires this resource. You could also see some ecological processes occurring. Some of them are sort of extensions of the evolutionary processes. For example, you might have shifts in the dominant species that have less need or improved uptake of that limiting resource. But also importantly, you could have the presence of new functional types. And here, most notably, for example, if there's a lot of nitrogen stress, you might see the appearance of nitrogen fixers. So they have sort of new functions. And so these short-term nutrient addition experiments may not capture the impact and trade-offs of such biological processes. Just to give you an example, if a cell is limited by phosphorus, it might start to produce something like alkaline phosphatase. Actually, many phytoplankton greatly express alkaline phosphatase when they're P-stressed, to the point where maybe half of their nitrogen in the cell is made up of this one enzyme. And so that's a hugely demanding process for nitrogen. And so they might actually start to run out of the nitrogen as they're trying to acquire phosphorus. And so then you're going to scratch your head, is this cell now limited by phosphorus or is it limited by nitrogen? And there's no real good answers to that. But of course, what started this whole process was potentially, in this example, the stress for phosphorus. What I hypothesize in this talk is that we can use the genomes and the functional traits, as well as the community composition of marine organisms to tell us about these biological processes. If an organism contains or expresses some of these different enzymes, right, it reflects that they're working hard to acquire a particular resource. Or if they've lost a lot of genes encoding proteins that uses, for example, a lot of iron or a lot of phosphorus, right? We can see that they're trying to shed or lower the demand for a particular resource. And so organisms and their ecology and evolution tells us a rich story about the stress that they are experiencing in their environment. So please hold that thought, because now I'm going to take a little detour, and then we're going to take a deep dive into this issue, because I would like to introduce the star of the show today, and that is Plecorococcus. That is my favorite organism that I have studied for most of my career. So Plecorococcus is a small marine unicellular cyanobacterium, and it is very small. So small that it took quite a long time before we realized it even existed, because it was evading standard techniques for detection. But now we learned that it's the most abundant photosynthetic organism on the planet, and there's more than 10 to the 28 cells of Plecorococcus in the ocean. It fixes around 5 petagrams of carbon, or nearly half of the total emissions from humans. It is present in most nutrient-limited environments. Because it's so small, it has some advantages in this type of environment. And as you can see on the map, it is really quite abundant from basically 40 north to 40 south. And lastly, and this is an important detail for the latter part of my talk, it grows very fast. It actually divides once a day, and this leads to a rapid selection for different genotypes. We know actually a fair amount about the biology of Plecorococcus, because it's so important for ocean processes. It's been the topic of quite a bit of study. And so through sort of a variety of work in the laboratory, as well as in the field, we can see that Plecorococcus has sort of functional traits that divides it into sort of 2 major groups. One is a highlight adapted Plecorococcus that grows near the surface, and another one is a lower light adapted ecotype that grows deeper in the water column. And as you can see sort of on the figure on the right, there's very strong selection for each of these genotypes, whereby the genotype that allows it to grow on the highlight is very frequent near the surface, and the genotype that enables it to grow deeper in the water column is heavily selected for deeper in the water column. When we isolate Plecorococcus and sequence its genome, one thing that we've learned is that among Plecorococcus, there might be sort of roughly 1,200 genes or so that are shared among all the strains. These encode sort of central metabolism, things that you need, that all cells need. But then, in addition, we see that there is a lot of genes that are ancillary to Plecorococcus, that are basically gained and lost between different Plecorococcus cells and genotypes. And so the Plecorococcus collective that grows out in the ocean basically consists of a multitude of unique genotypes. And these individual genotypes are then selected for under specific environmental conditions. During my postdoc, I did a fair amount of work on understanding how Plecorococcus adapted to differences in phosphorus availability. And so we grew Plecorococcus under low phosphorus and high phosphorus environments. And what we saw was that most of the genes that was preferentially upregulated when they grew at low phosphorus were all these genes that were involved in either the uptake of inorganic phosphorus, things like inorganic transporter, as well as genes that allowed it to access phosphorus bound to organic molecules, like alkaline phosphatase. And these all sat in one large cassette in the middle of the genome. However, when we sequenced genomes of other Plecorococcus cells, we saw that there was a lot of variability in the genome of these phosphorus acquisition genes. So for example, the top one, MET4, was the one we did the original experiment at, but its closest relative, which is more or less identical in the 16S ribosomal RNA, lacked all these genes. Then we had another strain, MIT 9312, which now had more or less the full cassette, but its closest relative, AS 9601, again lacked these genes. So there was a lot of variability even among organisms that phylogenetically was extremely closely related. Also, what we found was that the cells with the full cassette were typically, or the culture with the full cassette was isolated from a low P environment, whereas the cell from a high P environment lacked these genes. And so it appeared that there was an association between the environment of the origin and the presence of these genes. And this is something that we've confirmed in later experiments, that the presence or absence of these genes represents an adaptation to P stress. All right, so now we went to the field and looked at what is the frequency of these genes in natural Plecorococcus populations. And so the way we do this is that we sequence basically all the DNA that's in a given sample, and then we calculate the ratio of a particular Plecorococcus gene against the average presence of sort of core genes that we know that all Plecorococcus needs. And so if this population frequency is 0, it means that more or less no one cells in that population has this gene. And if the frequency is close to 1, it means that most cells in this population has a particular gene. And so as you can see on the figure here, the Plecorococcus population in the Sargasso Sea contains most of these genes. Also in the Gulf of Mexico, if we take a sample from there, we can see that this population also contains most of these genes. And the Sargasso Sea and Gulf of Mexico are notoriously low P and P stressed environments. However, if we go to a place like the equatorial Eastern Pacific Ocean or the central Indian Ocean, where there's more than 100 times the phosphorus as you see in the Sargasso Sea, we can see that the population, the frequency of these P acquisition genes in these populations are very different. As a matter of fact, there's been selection against most of these genes in these populations. And so it appears that there is a correspondence between the environment that these cells are growing at, whether it's high or low phosphorus in this case, and the presence of these different genes. We've also seen this for other elements like nitrogen and phosphorus. For example, with nitrogen, we see that there are different populations that either only uses ammonia or ammonia and urea, or ammonia, urea, and nitrite, or ammonia, urea, nitrite, and nitrate. So that's sort of a sequential. And we think that has to do with as nitrogen gets more and more oxidized, and thereby have a different oxidation state as the nitrogen that you need in your cell, it becomes energetically less and less favorable to use that compound as your nitrogen resource. For iron, we see that they might have siderophores to increase the uptake of iron, or they might lose various proteins that are sort of linked to the photosystem but not part of the core photosystem, and thereby lower their iron quota. Okay, so here we propose that we can use the gene frequencies of these different nutrient assimilation genes as a biosensor for ocean nutrient limitation. So we have sort of like this Plecorococcus collective of cells of all kinds of different genotypes. They get dispersed with the currents throughout the ocean, but then locally you have selection for different genotypes. So for example, if you have nitrogen limitation, you will have strong selection for cells that has a wide capacity for taking up various forms of nitrogen. If you have phosphorus limitation, like in the Sargasso Sea, then you have selection for organisms that can either have improved affinity for phosphorus or maybe accessing alternative forms of phosphorus bound to organic molecules. And the outcome is then when we analyze the populations that there are differences in the gene frequencies reflecting the environment that they're growing under. And the detail that is important to keep in mind is that because Plecorococcus grows so fast and you have such strong selection for different genotypes, then you get a very strong correspondence between the genomic content of the cell and the local environmental conditions. So now I'd like to present an implementation of this idea. And this is from a study that Chris alluded to that was published earlier this year, led by my grad student Lucas and postdoc Elise. And basically over the last 5 years, we've gone to a lot of different ocean regions. And I'll talk a little bit about this effort at the end of my talk. And there we sampled about 1,000 different stations from all the different major ocean regions. And so basically what we do is that we filter about between 2 and 10 liters of water, collect all the cells, we extract the DNA, we sequence the DNA, and then there's a lot of bioinformatics on the computer afterwards. But for this talk, what we did was we estimated basically the population frequency in Plecorococcus of these genes that are involved in nitrogen, phosphorus, and iron acquisition. And we had quite a bit of biochemical knowledge about these different genes. For example, this idea of nitrogen, the more oxidized the nitrogen is, the more energy it requires to assimilate it. And therefore, we could also use that biochemical information to assign these genes a priori to an understanding of, is this a severe stress, a high stress that the cells are experiencing? Is this sort of a medium stress, sort of maybe if they're only using urea, or is it low stress, they're just relying on ammonia? All right, so here's from one of the cruises that we went on. This is one of the AMT transects that maybe some of you are familiar with, that goes from the UK down to the Falkland Islands. And so on the left, we have samples from the Southern Ocean, and then we're basically going north up to North Atlantic temperate ecosystem. Okay, and in the red or pink, these are the frequencies of various iron acquisition genes. In blue are the frequencies of these different biomarkers I talked about before, these that you saw before, related to phosphorus acquisition. And then lastly, below is where you have in sort of orange is all the genes that are involved in nitrogen acquisition. And so let me highlight a few patterns. So first of all, there's been quite a bit of debate of whether or not cells are experiencing ion stress in the mild upwelling that occurs in the Atlantic. But we clearly saw indications of a high frequency of things like siderophores and other ion acquisition proteins in populations, and mainly in the equatorial Atlantic Ocean, where there is some upwelling, but not as much as in the Pacific. In the North Atlantic subtropical gyre, we saw clear evidence of phosphorus stress, as been proposed in many past studies. But interestingly, in the core of the North Atlantic gyre, we also see that there's a lot of nitrogen stress. So at sort of the edges of the gyre, they're mainly just P stressed, but in the core of the gyre, they're N and P co-limited. Lastly, we see very little stress in the Southern Ocean, as we would expect from the increase in macronutrients from upwelling. So we now have this type of data from all major ocean regions. Here's another example from a cruise that went from San Diego down to the ice edge of Antarctica. This is the P18 Clivar section. And some of the things that we see are very much what we would expect from past knowledge. For example, we see that there's very strong ion stress in the equatorial upwelling zone, and no phosphorus nor nitrogen stress in this place. But it's nice to see that our genomic indicators also tells us that there should be ion stress in the equatorial Pacific. However, what we see is then on the edges of the equatorial zone into the north, we sort of skirt the North Pacific subtropical gyre, and certainly go through the South Pacific subtropical gyre. And these gyre regions are nitrogen stressed ecosystems. But if we go and take a closer look at this type of data, we can also see how they're shifting resources as they're going along. So in the upwelling zone, that's what you see with the nitrate, the blue line here, right? That indicates where we have upwelling, and we have very little nitrogen stress when there's upwelling. However, what we can see is that within the gyres, both to the north and to the south of this upwelling region, the cells immediately, as you get outside the upwelling zone, starts to use urea, and therefore show some nitrogen stress. And then in the core of the South Pacific subtropical gyre is where they then go to the most difficult resource for them, which is nitrate. And so some of the things that we can learn from that is that urea, a reduced form of nitrogen, is used broader than nitrate, which is in a more oxidized form. And we can also see that the presence of these genes are not driven by where you have nitrate. So you could also think that in places where nitrate is available is also where they're assimilating it. But for Plecorococcus, at least, it's driven by the resource demand and not the supply, and thereby reflecting the stress that they're feeling or experiencing, I should say. All right, so now I want to show you sort of the synthesis of all this. If we do sort of a principal component analysis of these genes, we can sort of roughly divide the samples into populations that has a profile with a high presence of these P stress genes or N stress genes or the ion stress genes, or maybe a mixture of the two, showing some kind of co-limitation. And that's what you see in this plot. And so if we look at it now from on a spatial geographical scale, using this genomics assay, we can basically map out the global variation in the different types of nutrient limitation. And some of the things confirms patterns that we would expect based on past research. For example, the presence of a lot of phosphorus stress in the North Atlantic, iron stress in the Eastern Equatorial Pacific, and widespread nitrogen limitation. But what I think we're also learning from this plot and in this type of analysis is that there's also, for example, other zones of P stress and P limitation. For example, the western side of the South Atlantic subtropical gyre, which actually has sort of somewhat low phosphorus, and the cells indeed also experience P stress. We also saw quite a bit of P stress in the North Indian Ocean as you enter the Bay of Bengal. This was very surprising to us, but we've now learned that that is due to a very strong eddy field that occurs there. And so when eddies lead to downwelling, this is the situation where we can often see P stress. Other things that we have observed is that similar to some of these bioassays, we also see that on the edge of the subtropical gyres, as you're getting into more temperate ecosystems with a higher supply of nitrogen and phosphorus, we often see iron stress occurring. And so maybe iron stress is more frequent than just in the Southern Ocean and in some of the upwelling zones. Maybe it's sort of a common phenomena as soon as you start to have a presence of a significant supply of nitrate and phosphate. Lastly, in many places where we have nitrogen stress, we also often have iron stress, and I'll come a little bit back to that in a second. So we are collecting more and more DNA samples, both my lab group, but like lots of lab groups across the world. And by using this type of analysis, we can basically identify what are the limiting nutrients in a particular environment through bioinformatics. And I'll argue that that is a lot simpler and more quantitative than some of these bioassays, and can be done and really be scaled up. The other thing I wanted to talk about is that now that we can identify which regions have different types of stress, it has also allowed us to start speculating why it is that we have different nutrient stress types in different parts of the ocean. And we think that building on lots of past work in this area, we propose that basically when you have upwelling of nutrients, right, the nitrate to phosphate ratio in nutrient water that comes from the deep typically has a ratio of around 15. But in most surface regions where Plecorococcus is found, the cellular demand for nitrogen and phosphorus is much higher than 15. The other thing that is worth noticing here is that the iron content of deep water is often quite low. And so when you have this upwelling water, as has been demonstrated in many past studies, in the upwelling zones, you run into iron limitation first. And that is because you have basically a low iron to nitrogen ratio in this water coming in. And the dust deposition from the air that has some iron in it is not enough to make up for the low iron in the deep water. So stoichiometrically, you run out of iron. However, as this more water is now flowing away from the equatorial zones and towards the oligotrophic gyre systems, right, we have some dust deposition with iron. And so the iron to nitrogen ratio is increasing. And therefore, we often see in the boundary regions around the equatorial upwelling zones, we see sort of iron nitrogen co-limitation. As the water is flowing even further north or away from the upwelling zones, again, the iron to nitrogen ratio is increasing, and this iron is stimulating nitrogen fixation, adding new nitrogen into the ecosystem. And so, however, so what we experience here is generally nitrogen limitation. In places then where you get quite a bit of iron coming in, or a very low supply of macronutrients, and you then have high iron to nitrogen ratio, then you also stimulate the nitrogen fixation very strongly, and you basically push the system into P limitation. And so if we go back to our map from before, right, in places where we have a high vertical supply, a low iron to nitrogen ratio, like the equatorial Pacific, that's where we get iron stress. As we go away from this, so for example, the northeastern Pacific Ocean, or then you have an intermediate vertical supply, and you get sort of nitrogen iron co-stress. Another situation where you get nitrogen iron co-stress is in, for example, the South Pacific subtropical gyre, where you have a very low vertical supply of nutrients. It's very stratified, but you also have very low dust deposition, and therefore what we see is sort of nitrogen iron co-stress from this situation. But then as you get more dust deposition and some nitrogen fixation occurring, for example, in the South Indian Ocean, that's when you get nitrogen limitation. And then you have the situation with very high dust deposition, like in the Atlantic, high nitrogen fixation, and then P stress. So we also compared our work to these prior bioassays, and in general, there's a very strong correspondence between what we see and what they have found with this compilation of bioassays. So when a bioassay shows iron stress, we generally also see a high frequency of these iron stress markers, biomarkers. Same thing with PBOT when you add phosphorus or when you add nitrogen and stimulate growth. So there's a good correspondence. But the bottle experiments that you see on the right there represents maybe 20 or 30 years of work, and there's about 80 samples. The metagenomics represents about 5 years of work, and we have about 1,100 samples. And so it shows how we can scale the metagenomics way of understanding nutrient stress much easier than we can scale these bioassays, which are quite complicated to do. Another thing that we learned is that there's quite complex biology at these low nutrients. So as I said earlier on, that there was definitely correspondence between the P concentration and these P stress genes. And that's also what we see globally, that as the phosphate concentration goes down, in general in the global ocean, we also see an increased frequency of these P stress markers. However, what we also see both for N and P stress is that at the really low concentrations, there's a lot of variability in whether or not the cells show P stress or N stress. And what it tells us is that this genome diagnostic basically can capture that in environments, oligotrophic environments with really low concentrations, it's not clear whether or not that will lead to N or P stress. But having these genomic biomarkers, we can really delineate the type of stress that the organisms are experiencing in that environment. All right, now I want to shift a little bit and here towards the end and just introduce what we call Bioguruship. So some of you, or maybe many of you, are familiar with the Guruship program. This is sort of the extension of the old World Ocean Circulation Experiment, the WOS. And basically, Guruship is an effort to every decade cover most, if not all, ocean regions and measure things like the heat uptake or the uptake of anthropogenic carbon, or changes in circulation or changes in oxygen, things like that. And so every decade, this Guruship program covers most of the ocean and takes these chemical and physical measurements to understand the ocean as it's changing due to human activities. So what we are starting to develop is to add biological measurements to the Guruship program. And what I've tried to show today is how we can use, for example, some genomic measurements to get a deeper understanding of some biogeochemical features, in this case, what resource is limiting phytoplankton growth. And so more broadly, the vision for Bioguruship is to develop a deep understanding of the link between the physiochemical environment, which has been the traditional goal of Guruship, and the global ocean plankton biodiversity, abundance, and biogeochemical roles, all in the context of a changing ocean. And the idea is to leverage the global reaching Guruship platform and use these amazing measurements of the physical and chemical environments and link that up to this new set of biological measurements. And so, so far we finished 5 sections. That's the core of the data that I've shown today. And we've done sort of genomic sequences on all these different sections, as well as a variety of other types of measurements. We recently got funding from NOAA to do 3 more sections over the next 2 years. Those are the ones in yellow. And then hopefully within the next 6 years, we'll have finished all the sections that you can see in black. And so, and thereby starting to get sort of a truly global and at a fairly high resolution description of a variety of biological processes and link that up to our understanding of the physical and chemical environment. The people that are involved in this effort, myself, but also Harriet Alexander, Sophie Clayton, Jason Graff, and Nicole Polton, we're sort of the science leads in this. And then Heather Benway, Emmanuel Boss, and Luke Thompson are collaborating with us. And then, of course, also many junior researchers in our labs are working very hard on this project. And the idea is to use a variety of a combination of tools. Today, I've talked a lot about how we can use omics to understand ocean biogeochemistry, but we're also planning to use biooptics to get a more continuous description of various aspects of plankton communities, for example, the particle concentration, particle size distribution, the contribution of phytoplankton to total biomass and things like that. Imaging and flow cytometry to really get a detailed quantification of the abundance of different types of populations. And then finally, chemical measurements of leading to a detailed characterization of, for example, the elemental composition of plankton communities or their pigments. So some of the ongoing Bioguruship projects, so Chris alluded to one of them that I presented on at ASLO, where we are looking at the global distribution of diacetyl, so nitrogen fixers. We're also trying to develop a global understanding of distribution of sort of richness of the community, the biodiversity pattern. Another project that's very close to my heart is the stoichiometry, especially the carbon to nutrient ratios of plankton communities, and thereby linking together the supply of nutrients with carbon stocks and fluxes. And finally, to integrate this type of information to come and both evaluate climate predictions from the Earth system models, as well as come up with new theories for how we think plankton communities are going to be affected by climate change. Right now, a lot of Earth system models put a heavy emphasis on nutrient stress, leading to negative outlooks for things like productivity, phytoplankton biomass, and carbon export. But sort of this adaptive dynamics of communities that I've talked about today, how organisms adapt and rearrange in response to different nutrient stresses, may have an impact on some of these predictions for changes in productivity, biomass, and carbon fluxes to depth. And those are some of the questions that we aim to pursue. So to conclude, I would like to say that we can use the adaptive dynamics of microbial communities basically as a living biosensor for ocean processes. Today, I mainly talked about nutrient stress because that's something that I understand and have spent a lot of time on investigating. But one could apply the same things to heterotrophic processes, so the degradation of organic carbon in the ocean, or xenobiotics and pollution, questions of that kind, as well as impacts of slight changes in circulation on fields like oxygen, for example, that has a big impact on the biology of microorganisms. And the basic principle is that the fast growth and death of plankton in the surface ocean leads to very rapid selection and thereby a close association between the genotypes that are present in a given environment and the underlying environmental conditions that the cells are experiencing. I think one of the things that really changed over the last 5 years or so is that metagenomics has shifted from being this very exclusive, incredibly expensive, and very specialized tool to something that can be done really fast and relatively inexpensive, to the point where it's nearly as expensive for us to measure nutrients as it is to measure the full genome content of a population. And that has really, at least in my mind, changed the way that we can use metagenomics. And so, and under a relatively limited budget, do these large-scale characterizations of the genome content of plankton populations, and therefore really bring forward that type of information in the same way as we would use chemical measurements. We've also learned a lot about nutrient stress from the study. First of all, now, for the first time, we have a really global description of the differences in nutrient stress between different environments. And then we can start to ask questions about what is it that controls this biogeography of nutrient stress. And a lot of it is related to some ideas that have been presented in past research a lot, like the presence of upwelling and the role of iron and so forth. But what we can really now place that in a geographic context and understand how these differences in the supply of the major elements leads to sort of a local differences in which element is controlling plankton growth. And of course, here we've learned that nitrogen fixation plays a key role in tying together these three different elements. Finally, Bioguruship is an emergent project to quantify the global variation in plankton biodiversity, but really through that type of information that I've shown today, learn about the biogeochemical role of plankton in the current ocean, but certainly also how the ocean will change with future climate change. With that, I would like to thank you for your attention and take any questions.
Well, that was a great talk, Adam, and I'm gonna, as host, jump in and ask the first question, if you don't mind. And through your talk, I'm mainly thinking about this incredible genomic streamlining that occurs in Plecorococcus. That really facilitates a metagenomic approach for what you're doing. But I say that in the context of I guess I'm thinking about this approach versus a metatranscriptomics and the extent to which perhaps had metatranscriptomics been done, would the conclusions be any different? And then I'm also thinking, so that's like the first question. And the other one that I wonder about is, as obviously it doesn't get any smaller amongst the phytoplankton than Plecorococcus, but at some point you transition to a larger cell where there's not as much streamlining, and maybe the part of the ecological strategy is to have a broader tool set to deal with a dynamic ocean. And so, yeah, so those would be the two questions. If you did transcriptomics, how do you think things might differ? And then secondly, what would you think about it there being a threshold size, or maybe it's prokaryotic, eukaryotic, whereby you really you're better served focused on transcripts rather than genomes?
Well, I think there's obviously a long answer to that, and it's something that we thought quite a bit about. So first of all, one of the issues with using other omics tools like that are looking at expression with proteomics or transcriptomics is that there's a very strong diel cycle in these communities. So the cellular processes change rapidly from between day and night as they're expressing things, photosynthesis versus building new biomass and so forth. And so understanding the expression profiles, unless you really time it to the particular time of day and so forth, can be a bit messy. And so I think there's sort of a bigger question in that, which is that it provides sort of a time integrated view, and that time integration that we use for selection of different community members and genotypes maybe represents a time integrated view that is longer than the time integrated view of an expression, which might only be an hour or 2 hours or something, right? Maybe this is like 2 weeks. And so I think for the genomics that we're using, and so when we thought about how to do this study, right, we thought maybe we want to get not just sort of like the instantaneous responses, but more what is sort of the general sort of limitation that this community is experiencing sort of over at least more than a day, right? There could also be a little bit of drop of some nutrients or dead algae or something like that. And maybe that's not what we want to measure. We want to measure sort of an integrated aspect of that ecosystem. So the other aspect, and this time integrated view also plays into what about other organisms. And so larger organisms may grow longer, and the longer they grow, the more their genome will reflect the integration over that time scale. And so if you're in an ecosystem where they only divide once a week, right, then maybe the genomic content of that organism, and it takes a while to select and so forth, may represent a month or 2 months of an ecosystem. And so is that what you want to know? Then that's all right. If you want to know sort of more on a shorter time scale, then maybe you want to go to something like gene expression and so forth. So I think it's just a matter of thinking about the time scale for which you want the signal, and how does that time scale relate back to the biology of these organisms.
Well, that makes good sense. I'll just note that I will be departing shortly. Josie will be the host when I have to go, but I see our dean, Paul Shepson, has a question.
Yeah, your atmospheric science deans, but I'll venture in. That was just really cool talk. I'm really impressed with the metagenomics approach. I'm just curious if, given what you have learned and what is known about the factors that contribute to nutrient limitation, if you could use some form of ocean system modeling to, sort of retrospectively determine what that global pattern of nutrient limitation would look like prior to the Anthropocene. You have any sense of that?
Yeah, so we are working a little bit on, we were sort of more looking forward rather than backwards, but the questions are very similar, right? And so my colleague down the hallway, Keith Moore, he is deeply involved in the CSM, Earth System Model, and on the ocean side. And so we compared our maps of nutrient stress quantitatively to how his model predicts variability in nitrogen, phosphorus, and ion stress. And I think it's interesting what we see is that, for example, for the Atlantic Ocean, they match really well, generally, which probably reflects that we have a really good understanding of the biogeochemistry of the Atlantic Ocean, whereas the Indian Ocean was really crappy. And I think that also reflects the fact that there has just not been a lot of focus on biogeochemical processes in the Indian Ocean for geographic reasons, sort of lack of major funding source neighboring the Indian Ocean. And lastly, I think the Pacific Ocean, I think most Earth system models underestimate how much the biogeochemistry shifts as you go from east to west. And the sloping isopycnals that occurs in the in the Pacific Ocean really changes a lot of the biogeochemical processes. And some of this are not really well. So this interaction between the physics controlling the chain, the deepening of the isopycnals, and the effect on the biology, this interaction is not so well captured right now in Earth system models. And we have a paper a few years ago where we analyzed this in a little more detail. And I think, so coming back to your question, is that this interaction between the physics and the biology around the stratification, I think indicates that there are still some things that we need to add into the Earth system models in order to capture these biological responses. And I think it has a big impact on our estimation of changes in productivity.
Thank you.
So I see there's a chat question from Monica. If you're there, do you want to ask it yourself?
I think Maddie also had a question. Madeline had a question first, so I don't mind if she
Oh,
wants
okay,
to go.
I see Madeline. Okay.
Yeah, hi. I was just asking because I was thinking about like sort of the ability of like planktonic organisms to sort of have their DNA recombined by like viruses. So I was wondering, how do you think virology plays a role in genetic recombination in the case of Plecorococcus?
Well, I think it's clear that they do play a role. You know, I don't think we fully understand how this gain and loss of genes occur in these populations and how much it's sort of actively recombining versus that maybe it happened 1 million years ago and now you just have lots of different genotypes that are being selected as they get dispersed around with the ocean currents. And so the answer to your question, right? So a virus is sort of today important for the recombination, maybe. But I think also embedded in that question is how much recombination do you actually see, and what is the rate of recombination in the current ocean? And I think that holds up again sort of, well, how much is just selection of existing genotypes, because there's just a lot of sort of diversity of Plecorococcus that's sort of being slushed around. And so I don't know the answer to that. I think everybody would agree that over sort of a larger time scale, viruses definitely played an important role, but how much they sort of impact the rate today, I don't know.
Thank you.
Let's see. Monique, did you?
Yeah, I had a question for the Bioguruship Project. So it looks like you were talking a lot about for the genomic work and as well as looking at kind of biodiversity. It seems like you do a lot for microbes and phytoplankton. Do you also do work on zooplankton as well?
So, so there are two answers to that. The first one is about myself, and I don't do so much on that, and it's in part reflecting the fact that while that undoubtedly zooplankton are really important, I think it's still an unclear question of how zooplankton biodiversity affects these smaller phytoplankton. Right, we know they're going to get eaten by someone, but who that someone is and whether or not shifts in the zooplankton community affects the death rate of Plecorococcus, I think are still open questions. The second part of my answer would be that there is a lot of information in the data that we are collecting, right? We're collecting every organism. Now there's a numerical argument, so the ones that are really abundant are also going to be the ones that are well represented in the DNA. But that doesn't mean that there's not eukaryotic DNA present in our samples. It just requires that somebody goes in and start looking at it. And I think we don't have as many genomes for zooplankton as we have for some of the phytoplankton. And I think that sort of limits maybe what we can do with these metagenomic. But I think there's a big area for which a lot can be done.
All right, then, thank you.
Jeff Levington. Jeff, you're on.
Yeah,
unmute.
okay. So first of all, thank you. I think I'm saying for everybody what a wonderful seminar this was and how enlightening it was. There's something that interests me. It sounds like you're using, and I think it's a very exciting way to do this, to use expression, gene expression as a bioassay. And so I guess my question is, when does this go wrong? And so let me give you some specific questions. There are parts of the ocean, of course, where certain groups of phytoplankton are extremely important in processing nutrients. Diatoms, for example, in certain parts of the ocean, dinoflagellates, trichodesmium, and others. So I'm wondering if you've thought about whether there are specifics about the genomes and the genetic expression of these different groups that might lead you in different directions and even lead to different conclusions about which nutrients might be limited, just basic limitations that are different than they might be for Plecorococcus.
I think that's a great question, and we have thought a little bit about it, and we recently received some new funding to maybe push that a little bit. So I think the limitation, right, is in part driven by the balance between the supply and your demand, right? And so there's a supply N to P to FE ratio in the supply, as I talked about today. But of course, there's also the N to P to FE ratio of your demand, right? And in the case where, for example, a diatom may have a different demand than Plecorococcus, right? And so let's say that it needs more nitrogen just for the sake of it, right? But then you could have the situation where Plecorococcus may show phosphorus or iron stress, but the diatom may show nitrogen stress because it has a different demand. And there's actually some precedents for this idea. Harriet Alexander, who's also part of Bioguruship, she did some really nice studies during her PhD, I believe, where she showed that a dinoflagellate and a diatom, I believe it was, coexisting. One was limited by nitrogen and the other one was limited by phosphorus, even though they saw the same environment. And so I think we've also done some work on the elemental stoichiometry of different lineages. And we generally see that Plecorococcus uses a lot less phosphorus than maybe some of the larger eukaryotes. And so I would expect to see that Plecorococcus could be nitrogen limited and some of the other ones could be phosphorus limited in the same environment, just as an example from that. And so I think it's super interesting to start to think about, okay, this is just Plecorococcus, but what about the other members? And I'm sure there's going to be variation in the stress profile. And I think it will be great to really learn about that. And I think this is just the beginning. And I think, but now maybe we have some tools whereby we can ask these questions, which we couldn't do when we were just doing bioassays, or at least it was harder. Sometimes people looked at what was growing, but still, this one, I think, with these types of genetic tools, I think we can really take a deep dive into that. And I'm sure we're going to see some interesting biology.
Thank you. I would add also that along with physiology, there might be different weirdness components of the genomes of these different phytoplankton groups. I seem to remember that trichodesmium has a really weird genome that bears some scrutiny about nutrient uptake. So there might be some other examples like that. Yeah.
And of course, the nitrogen fixes, right? They are sort of nearly by default, right, not going to be N limited.
Exactly.
Right, and so they're for sure going to be somewhat different from Plecorococcus.
Thank you very much.
Gordon.
Hi.
Gordon
Adam.
Taylor.
Thanks for that wonderful talk. Really enjoyed it. And I want to commend you on the scale of this project. It's really impressive. But we have to bear in mind that you're taking snapshots at one time of year. Would you get a different answer in temperate environments if you went 4 times a year, for example?
I'm sure to some extent. Now, Plecorococcus is probably a good choice for this because most of where it is, like they're mostly in fairly stable environments with not a lot of seasonality. I mean, I'm not saying no seasonality, but not a strong seasonality as maybe some of the higher latitude environments. But one of the things that we are starting to look at is, for example, at bats, where we have Plecorococcus year round, but there's quite a bit of difference in the nutrient supply example with the winter upwelling, winter mixing and such. And so there you could easily imagine that in the summer, fall stratified season has very different nutrient stress profile than February, March. And as you go further north, I'm sure that the seasonality will be stronger and stronger. Now, I will say that my lab and other labs have gone to the Western North Atlantic now on 5 different occasions and done metagenomic sequencing. And each time, it's always coming out like a strong P stress in the genome. And so you're totally right that it is just a snapshot. And I think a snapshot maybe that tells you something about the last week or two. But there is also stability in these types of ecosystem, many of these ecosystems, that sort of makes it probably reasonable to think about a large scale biogeography. But of course, recognizing at some point we got to do the globe and variation in time, but that's of course a much larger project than just the globe itself.
Yeah. So with the Bioguruship project that's gearing up, your list of variables, they seem to all be sort of state variables. There's no, I didn't see anything on rates. Is that just too ambitious to do on the Bioguruship?
Yes, it's a great, it's a sole point in a way. I think my thinking was similar to the regular Guruship program, right? Is to start out with a focus on the state variables and then use modeling, whether it's forward or inverse modeling, to start capturing some of the rates, or at least try to see what rates would explain the stuff that we're seeing. But ultimately, right, I would love to start to see more rate measurements, but there's a logistical constraint on how many things. The physical oceanographers are already sort of complaining that we're encroaching on the water and the birth and so forth, right? So maybe we shouldn't be too greedy to start out with.
Yeah, great. Thanks very much. I see Ben has a question.
Yes, and just to kind of copy everyone else, that was a really great talk and really interesting findings. I think one of the biggest question that came to mind for me was, in these parts of the ocean where you see high iron and low nitrogen levels, you see an increase in nitrogen fixers and nitrogen fixation. I was wondering if there's anything that you've seen in your work or other people have done which show whether nitrogen fixation can be really effective and really efficient in supporting microbial communities, or does it kind of just sort of maintain microbial communities as they are, just trying to survive under these relatively low nitrogen conditions?
Well, so this is probably a question that's beyond my own data, but I can give you my thoughts. Right? I think it's a tight dance between in many of these ecosystems, Plecorococcus and the nitrogen fixers. In that Plecorococcus, as we have seen in other work, also from Bioguruship, has quite variable stoichiometry, and they can use up to maybe 40:1 nitrogen to phosphorus in the biomass, and so a happy Plecorococcus cell will use a lot more nitrogen than what is provided by the flux from the deep ocean, and this opens up a niche for nitrogen fixers, as the rest of the community is nitrogen stressed. They have sort of their own supply and therefore can invade, despite that they're growing slow and have to use so much energy. And so I think if we sort of think about it from the perspective of Toby Terrell or someone like that with his models, right? I think what they're doing is that the phytoplankton wants more nitrogen to phosphorus than what is being supplied. The nitrogen fixers can provide that, and therefore both of them can coexist. But the big sort of extra variable is then whether there's enough iron to support that additional nitrogen fixer activity, and that's what then ultimately leads to the control on nitrogen fixation in the global ocean, is whether or not there is enough iron compared to nitrogen for that niche. So I don't know if that totally answered. So I think they're both dependent on each other, right? Plecorococcus wants more nitrogen, and the nitrogen fixers from that want of Plecorococcus opens a niche for the nitrogen fixers. So I think they sort of go hand in hand.
Yeah, absolutely. Thank you.
Any other questions? Okay, well, thank you very much. That was a very, very interesting talk, and I think we all really appreciate your willingness to come to us today.
Yeah, absolutely. It was great to present to you, and of course I know quite a few of you at you, and it was great to present this work.
All right, good. All right, if there are no other questions, people. Well, okay, well, thank you.
Thank you.
Bye.
Bye.
Bye.